Hello everyone, today's topic is nucleic acid. I will discuss this topic in details. It is the most important topic in biochemistry and as well as molecular biology. If you like this video, please like, comment and subscribe my channel. If this video is helpful to you, please share it with your friends and support my channel. So let's start. First, nucleic acid. What is the nucleic acid? The definition of nucleic acid. Nucleic acid are the large biomolecules which is composed of the 5-member ring sugar linked by phosphate group and 4 nitrogenous base. So, nucleic acid is a large biomolecules which is composed of 5-member ring sugar, phosphate group and nitrogen base. So when these three components are bind with each other, they form nucleic acid. This is the class of conjugate protein. Then what is the basic structure of nucleic acid? In a nucleic acid, a nitrogenous molecule which is attached with the sugar molecules, and one sugar molecule is attached with another sugar molecules by a phosphate group. As the same way, one nitrogenous base is attached with the sugar and sugar is attached with another sugar molecules with a phosphate group. As the same way, there is a lot of nitrogen base sugar, phosphate group, sugar, nitrogen base, and there is a n number of this structure can form nucleic acid. Then the component of nucleic acid. What is its component? The component of nucleic acid are sugar, nitrogen base and phosphoric acid. Sugar can be divided into two types. One is D-ribose sugar and another is 2-deoxyribose sugar. Then nitrogenous base. Nitrogenous base can be divided into two types, purine base and pyrimidine base, and another phosphoric acid. Then the first component is sugar. So first component is sugar. It is a 5-carbon sugar that is also called pentose sugar. Pentose sugar. Where pent means 5 and it is came from penta and tose means sugar. So it's called pentose sugar. Then the types of it can be divided into two types, D-ribose sugar and 2-deoxyribose sugar. D-ribose sugar is found in RNA and 2-deoxyribose sugar are present in DNA. Then this is the structure of D-ribose sugar. There is a oxygen molecules and there is 1, 2, 3, 4 and 5 carbon molecules. So it's called pentose sugar, 5 carbon molecules, and here there is a present an OH group. So it is the main causes of, it's called RNA, D-ribose. And the second 2-deoxyribose sugar, here you can see that 1, 2, 3, 4 and 5 carbon molecules and 5 carbon molecules, and here this is only H groups. There it is OH groups and here it is only H groups. So it's called deoxyribose. Deoxy means the removal of oxygen. So here the oxygen group is removed, so it is called 2-deoxyribose. Then the nitrogenous base. The second component is nitrogenous base. It is 1 or 2 ring sugar structure include nitrogenous atom. So it is there is a 1 or 2 ring sugar is present in the nitrogenous base. First, purine base. Here is a 2 ring sugar is present. So it's called double ring structure. Purine is the double ring structure. It can be divided into two types, adenine and guanine. Here it is a structure of adenine. You can see that there is a in each two groups in adenine, and in a guanine there is a O groups. Double bonded O is present in guanine. You can practice this structure. And this both of the structure is present in DNA and as well as RNA. Adenine and guanine means purine bases is present DNA and RNA. The second types of nitrogenous base is pyrimidine. It is a single ring structure. You can see that thymine single ring structure, cytosine single ring structure, and uracil single ring structure. It is three types: thymine, cytosine, and uracil. The thymine structure, there is a double bonded O, where in a cytosine structure there is a NH2 groups, and uracil there is O group. So you can see in the cytosine there is NH2 groups, and we also see there is an adenine groups where is NH2 groups. So cytosine and adenine where is present NH2 groups. And the other structure is almost same in three structure: thymine, cytosine, and uracil. And in the DNA there is present cytosine, and thymine. And in the RNA there is present cytosine and uracil. So cytosine, in the pyrimidine base there is a cytosine that is present in both structure DNA and RNA, but the thymine is present in DNA and uracil is present in RNA structure. Then the phosphoric acid. Phosphoric acid. It forms an ester linkage between two nucleotide on RNA and DNA. I will discuss after how the ester linkage can bond with two nucleotides in RNA and DNA. So this is the structure of phosphoric acid, where OH group is present and double bonded O. There is the O which can easily bind with H groups, and there is a two OH groups. Then nucleoside. Then nucleoside. So nucleoside is the combination of nitrogenous base and pentose sugar molecules through the glycosidic linkage. So when a nitrogenous base and a pentose sugar molecules are joined with each other or bond with each other through the glycosidic linkage, they form a nucleoside. You can see there is a adenine structure. It is an adenine structure, and it is 2-deoxyribose sugar, where there is the free OH molecules and there is an H molecules. They are bond with each other and form a glycosidic linkage. You can see that this structure and this structure almost same, only there is H and OH groups that can bond with each other and form a glycosidic linkage. It is a adenine structure, which is bond with the 2-deoxyribose sugar, and adenine and 2-deoxyribose sugar ribose sugar form 2-deoxyadenosine. So you can remember this. In a purine nucleotide, the C1 of the sugar, C1 means 1, 2, 3, 4, 5. So this is the first carbon. So in the purine nucleotide, C1 means carbon 1 of the sugar molecules are attached with N9. 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 nitrogen 9 of the purine bases. So in the purine nucleotide, the C1 means carbon 1 of the sugar molecules can attached with the N9, nitrogen 9 of the purine bases. This adenine is the purine bases. Then the cytosine is a pyrimidine pyrimidine nucleotide. So cytosine 1 2 3 4 5 6 and sugar 1 2 3 4 1 2 3 4 5. So there is a cytosine. In a cytosine, nitrogen, one number of nitrogen can attached with one number of carbon. It is a 2-deoxyribose sugar. You can see there is only H groups. If there is an OH group, it called D-ribose sugar. Ribose sugar. So it form an RNA structure. In this case, it is a DNA structure. So 2-deoxyribose sugar can bond with the cytosine though the glycosidic linkage. You can see that there is a one number of nitrogen can attach with the one number of carbon. So it formed 2-deoxycytidine. It is called when an cytosine group is attached with the 2-deoxy-D-cytidine, 2-deoxy-D-ribose, they form 2-deoxycytidine. In the pyrimidine nucleotide, C1 means carbon 1 of the sugar molecules is attached directly with the N1 means nitrogen 1 of the bases. Nitrogen 1 of the bases and C1 of sugar. Then the second and most important concept is nucleotide. What is nucleotide? Nucleotide, it is a combination of nucleoside and phosphoric acid. So nucleoside and phosphoric acid. Nucleoside is bound with bases and sugar. So when base, sugar and phosphoric acid 3 are bind with each other, they form a nucleotide. This base, sugar and phosphoric acid are the precursor structure of DNA and as well as RNA. DNA deoxyribonucleic acid and RNA ribonucleic acid. So in the nucleotide structure you can see that there is an adenine, a purine bases and a sugar molecules. It is a 2-deoxyribose sugar, or if it is OH group, so it form ribose sugar, D-ribose sugar. It is called DNA, and if it is OH group, so it is called RNA. But other the structure is same as well as in DNA and RNA. So here is a phosphoric acid which can attach with a sugar molecules. So in the nucleoside there is attached with bases and sugar, and in the nucleotide there is attached with a phosphoric acid. So you can see that this is the adenine group, that is 1 2 3 4 5 6 7 8 9. So N9 of adenine group or the bases are attached with the C1 of the sugar, and here this is the 5 number of carbon that can attach with the phosphate groups. So when an adenine group and 2-deoxyribose sugar and phosphoric acid bond with each other, they form 2-deoxyadenoside 5' monophosphate. Another and most important things is when then adenine and sugar base attached with each other, they form a glycosidic bond. And when a phosphoric acid is attached with the sugar molecules, they form ester linkage. So in DNA, a deoxyribose sugar can attach to a phosphoric acid to a free OH group present at C3 and C5. You can see this is a sugar molecules and H groups. It is a DNA sugar molecules. So there is a free OH group. This 1, 2, 3, 4, 5. So there is a free OH group and there is an other OH group, free OH group. So C3 and C5, there is a free OH group. So in DNA structure, a D-ribose sugar can attach to a phosphoric acid, to a free OH group, either C3 or C5. And as the same way, in a RNA molecule, the ribose sugar can attach to a phosphoric acid present at C2, C3 and C5 to form a ester linkage. You can see that in this if this is a D-ribose sugar, so in C2 structure there is an OH group also present, there is an OH group. So OH group is the free OH group that can also bind with the phosphoric acid. So in RNA structure, C2, C3 and C5, the phosphoric acid can bind with the sugar molecules. So in DNA structure, there is a sugar molecules where is present a free OH group in C3 and C5. In RNA structure, a free OH group is present in C2, C3 and C5 that can form a bind between phosphoric acid and sugar molecules and form a ester linkage. This is a chart where bases, nucleoside, nucleotide. Nucleoside is the base plus sugar, and in the nucleotide it is base plus sugar plus phosphate group. So we can read also that bases, there is a two type of base, purine and pyrimidine. Purine basis is adenine and guanine. Pyrimidine basis is cytosine, thymine and last uracil. So when adenine in the nucleoside, it is called adenosine. In case of RNA, if it is DNA, there is deoxyadenosine. Deoxy is the only one that is attached with the adenosine. So RNA and DNA, there is the same adenosine, adenosine. But in the case of the DNA, as there is a 2-deoxy-derivose sugar is present, so there is a attached deoxy word. And as the same way, there is adenine in the nucleotide is called adenylate in RNA and deoxyadenylate in DNA. Guanine, guanosine in RNA and deoxyguanosine DNA. In the nucleotide, guanine is also called guanylate in RNA and deoxyguanylate in DNA. Cytosine is called cytidine in RNA and deoxycytidine in DNA. As the same way, in the nucleotide, there is a cytidylate is RNA and deoxycytidylate is DNA. Thymidine or deoxythymidine is DNA, and thymidylate or deoxythymidylate both are called in DNA, as the thymine is only present in DNA. Then the uracil, that is present only in RNA, not DNA, and the thymine is present in DNA, not RNA. So in the cytosine, nucleoside is called the uridine, and in nucleotide is called uridylate. So in the nucleoside, there is a sine that is used as a suffix in adenine, guanine, cytosine, thymine or uracil. And in the nucleotide, there is added a late as a suffix in a purine and pyrimidine bases. The types of the nucleic acid depends on the number of nucleotide unit it can be divided into three types: mononucleotide, dinucleotide, and polynucleotide. Mononucleotide, that is composed of one nucleotide, and example is AMP, and adenosine monophosphate, adenosine diphosphate, adenosine triphosphate, ATP, AMP, ADP, ATP. And in dinucleotide, it is composed of two nucleotide unit. Example is NAD, nicotinamide adenine dinucleotide, and FAD, flavin adenine dinucleotide. In the polynucleotide, it is composed of the main nucleotide, and example is DNA and RNA. DNA is deoxyribonucleic acid, and RNA is ribonucleic acid. It also can be divided, depends on sugar molecules. Depends on sugar molecules. It can be divided into two types. This is a 2-deoxyribose d-ribose sugar, and d-ribose sugar. If 2-deoxy-d-ribose sugar is present in the sugar molecules, so it is called the DNA. And if the d-ribose sugar is present, it is called RNA. Then what is the function of nucleic acid? The function of nucleic acid is, first one, storage of the genetic information. It can store genetic information and can transfer from one generation to another generation. So the second point is transfer genetic information, that I say help in protein synthesis, act as a energy storing molecules, and play an important role in carbohydrate, protein, and fat metabolism. So that's all. So we can discuss about the nucleic acid. In the details of nucleic acid, you can see there is their definition, structure, component, what is sugar, what is the 2-deoxy sugar, what is, and their structure, nitrogenous base, purine bases, pyrimidine base, phosphoric acid, and what is its function. So I think this video is helpful to you. If you like this video, please like, comment, and share this video with your friend, and also subscribe my channel. Thank you everyone.